Good morning, everyone. Great to see everybody here bright and early this morning. I'm Janet Rossant, and I'm President and Scientific Director of the Gairdner Foundation. Welcome you all to the 2026 Canada Gairdner Awards announcement. Before we begin, we acknowledge that the Gairdner Foundation, and indeed Mars in total, operates on the traditional territory of many nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat peoples. And this land is home to many diverse First Nations, Métis, and Inuit communities. And as we celebrate scientific excellence today, we recognize the long-standing knowledge systems and innovations of Indigenous peoples on these lands. It's wonderful to be with you here today in person, and to welcome also those of us who are joining us online around the world as we reveal the recipients of the 2026 Canada Gairdner Awards. We're honoring exceptional scientists whose discoveries are expanding our understanding of biology and impacting human health globally. And at a time when trust in science cannot be taken for granted, it's more important than ever to recognize the individuals whose work drives progress and improves lives. The Gairdner Awards celebrate not only scientific breakthroughs, but also the global collaboration, curiosity, and dedication that makes those breakthroughs possible. We must, and we do, remain committed to supporting diverse voices in science and inspiring the next generation of researchers who will shape the future of discovery. Please join us in this quest. We're very proud today to say that the Power Corporation of Canada is the presenting sponsor of the announcement, and we're deeply grateful to their ongoing commitment to celebrating excellence in science. We also thank our media partner, I'm just checking the slides here. We thank our media partner, the Globe and Mail, for helping share the stories of our laureates with audiences across the country. The Gairdner Foundation, for those of you who are not familiar, but I think many of you are, was founded in 1957 by Canadian philanthropist Jim Gairdner to recognize researchers whose discoveries have transformed our understanding of biology and impacted human health. Over the years, the awards have earned global prestige and are a Canadian icon, on a par with the very best awards in the world. To date, 443 scientists from over 40 countries have been honored, including 103 who went on to receive a Nobel Prize. We're very happy and honored today to be joined by 3 government leaders who share our vision of advancing research and innovation in Canada. We're very pleased to welcome Maggie Chi, Member of Parliament for Don Valley North and Parliamentary Secretary to the Minister of Health, and Karim Bardezi, Member of Parliament for Tayayokan, I can't pronounce it, I'm sorry. Parkdale, maybe when you come up, you can say it for me. It's a very difficult one. And he is Parliamentary Secretary to the Ministry of Industry. I would like to acknowledge the long-time support of the Government of Canada and the Canadian Institutes of Health Research, whose partnership has helped advance the Gairdner Foundation's mission of celebrating excellence in health research and sharing discoveries with communities across Canada and around the world. Their financial support also made it possible to increase the value of each Canada Gairdner International Award to $250,000 beginning last year, so it's a really important contribution and change. We're also honoured to be joined by the Honourable Nolan Quinn, Ontario's Minister of Colleges, Universities, Research Excellence and Security. And we're very grateful to the Government of Ontario, who have recently renewed their support for the Gairdner Foundation. And that support will help strengthen the pipeline of young scientists and entrepreneurs in Ontario. Together, we will ensure that the province is firmly on the global map for scientific excellence. We're going to hear from each of our government partners, but since Minister Quinn has another appointment soon, I'd like to invite him to the stage now to share a few words. Minister Quinn.
Thank you for the introduction. Good morning, everyone. It's a privilege to be here to celebrate the achievements of exceptional researchers whose discoveries are not only advancing life sciences, but improving lives here in Ontario, but also across the world. For more than 65 years, the Gairdner Foundation has championed scientists whose work is shaping our collective future. By celebrating scientific excellence, the Gairdner inspires the next generation of scientists who will secure Ontario's position as a world leader in discovery and innovation. The fact that more than 100 Gairdner Award recipients have gone on to receive Nobel Prizes speaks volumes about the caliber of this recognition and the lasting impact of the work being celebrated here today. This year's recipients exemplified the best of research excellence, driving discoveries that are changing lives here and across the globe, which is exactly why our government is proud to invest in homegrown research. Because we know an investment in Ontario-made innovation is an investment in a brighter, more prosperous future for our province. Since we have taken office, our government has invested more than $2 billion in the research sector, supporting more than 1,800 projects that are pushing the realms of what is possible across critical fields like advanced manufacturing, mining, and of course, the life sciences. And to further this work, just last year, as was just announced, a partnership with the Gairdner Foundation providing $600,000 to raise the profile of life-saving research like the work being honored here today. So to the winners, congratulations on your remarkable research. Thank you for advancing health and improving the lives of the people of Ontario and around the world. Our future is very much brighter because of your curiosity and your dedication, and our government is proud to support you. Thank you, and again, congratulations to all the winners today. Thank you.
Great. Thank you very much, Minister Quinn, and for your continued support of research and innovation in Ontario. So now please join me in welcoming MP Maggie Chee to share a few remarks from the Ministry of Health of Canada.
Thank you, Minister Quinn. I think you just left. I'm pleased to be joined here with my colleague, Parliamentary Secretary Karim Bardizis as well. Thank you, Dr. Rossant, and greetings, everybody. Bonjour tout le monde. What a wonderful day for science, everyone. I'm delighted to be here representing the Government of Canada. It is a pleasure to join Minister Quinn and other dignitaries for today's announcement of the 2026 Canada Gairdner laureates. In Canada, as Dr. Rossant has repeatedly said, we believe in science. Scientific discovery have increased the human lifespan, improved our quality of life. Research helped deliver new treatments for major diseases such as cancer, heart disease, and diabetes, and has ended global epidemics such as polio. Every day, scientists are making groundbreaking discoveries, improving care for patients and saving lives. As a country, we recognize that science is key to building a strong, more resilient, and healthier Canada. The Government of Canada is proud to support the Gairdner Foundation. Gairdner strengthens Canada's position as a global leader in health and life sciences by convening the world's top researchers and celebrating groundbreaking breakthroughs here at home. Gairdner's extensive outreach connects laureates directly with thousands of Canadian students each year through Gairdner's Science Week and year-round programming. These interactions inspire the next generation of scientists and innovators, helping to build a strong, diverse pipeline of scientific talents that supports the long-term health and competitiveness of Canada's life sciences sector. To you, Dr. Rossant, and everyone at the Gairdner Foundation, thank you for everything you do to promote Canada's leadership in recognizing the transformative discoveries that improve health globally. And to the 2026 laureates, congratulations on receiving this international recognition for your pioneering research. Your discoveries have made long-lasting contributions to scientific research, and they will have an enduring impact on the health of people in Canada and around the world. And I urge you to keep pushing forward because there's still much to be done. in science, when one of us succeeds, everybody benefit from that success. Thank you so much, everyone.
Thank you so much, Ms. Chi. That was fantastic words that we all live by. And now I'd like to invite Karim Bardisi to say a few words.
Janet. Hi everybody, bonjour, je suis Karim Bardisi, le secrétaire parlementaire de la ministre de l'Industrie, l'honorable Mélanie Joly, et aussi le député de Taillé-Con Parkdale High Park. And it's a great pleasure, Maggie and I get to participate in these events from time to time because of the partnership between Health Canada and Innovation, Science and Economic Development Canada on research. And in this moment, in this event, in this foundation and its awards, we are standing on the shoulders of almost 70 years of a really good idea that then we all get to come in behind. And I'm so appreciative that we have these partners that have come in to help support that. And I think maybe one of the original insights from the foundation's founding is that we shouldn't shy away from excellence. And that excellence can be found even beyond our borders. And by recognizing excellence beyond our borders, it enriches us. I think our government has taken that on board a little bit. Maggie mentioned our belief in science at a time when others are not believing in it so much. And so that has been the inspiration, for instance, for the creation of the Impact Plus Chairs program. So I don't know if John or Jeff are thinking of spending more time in Canada in the coming years, but we know that we put a really major investment after the 2024 budget, which made major investments in the granting councils that support hopefully some of the people in this room, a 2025 investment that's going to bring over 1,000 researchers of all tiers to Canada or back to Canada to do the next generation of work, to work on the discoveries and the insights that Maggie was talking about. Being at an event like this is a specific pleasure because, we fund the ecosystem of research, but there are these special investments that we as a government do that really highlight the potential of scientific research. And I'm thinking of Genome Canada that maybe some people here are involved in, where policymakers of the past have decided, we really need to focus on this one area and really lift up the mission-driven research here. And in the Canada Gairdner Awards, we see a bit of the result of that. I also just want to share my thanks to Janet and the chair and Heather and the people who have made the very important insight that intergenerational outreach is a good thing, and that if we are going to celebrate science, we have to reach deep into the next generation of science, non seulement dans les universités, mais même dans les écoles secondaires primaires, that we have to go really deep into the ecosystem and find those people who don't know they want to be scientists, but with the right kind of inspiration. And I know Gairdner Foundation shares as a peer with organizations like the Perimeter Institute and others that are elevating excellence on the one hand, but also pulling in the next generation of researchers. So with Maggie and on behalf of Mélanie Joly, I'm very thankful for the opportunity to share this with you today and to share our partnership with you. Thank you.
Thank you so much, all our government partners. I think it's wonderful to be able to get together and share our common goals. We have common goals, and they are goals that are shared across the country. So Canada stands for science, and our government partners stand for science, and that's a wonderful message for all of us to hear. I'd also like to just welcome Paul Genet from Power Corp. We thanked Power Corp for sponsoring the event today, but now you're here, I'd like to recognize you personally. So thank you, Paul, for your ongoing, long-time support for the Gairdner Foundation. So we're getting close. We're getting close to the moment that if you haven't found the rumors that are out there, getting close to the time when we're going to announce the awards. But before I do that, it doesn't just happen overnight. We don't just pull names out of a hat. There is a long, complicated process that leads up to the selection of the laureates. And I'd like to recognize at this point the extraordinary work that goes into selecting the year's laureates. We have a rigorous multi-stage evaluation process that involves leading scientists from Canada and around the world. And I'd like to extend my sincere thanks to members of our medical review panel, the medical advisory board, and the adjudication committees for the Global Health and Momentum Awards. Their dedication and thoughtful deliberation ensures that the Gairdner Awards continue to honor truly outstanding scientific achievements. And the rigor of our adjudication process is really what is recognized internationally as being something that puts Gairdner right at the forefront of these kind of awards. I, of course, would also like to acknowledge the leadership and support of the Gairdner Foundation Board of Directors, led by Dr. Heather Munro-Bloom, Chair of the Board, along with our board members, several of whom are joining us today in the room or online. Your commitment to the foundation and to our mission is deeply appreciated. Okay, now, without further ado, it's time to announce our laureates. I just want you to know that we actually have 3 laureates in the room, and the rest will be joining us online for a discussion at the end of the process. So you're going to meet some in person, you're going to meet some online, so they're all with us today. So I'd like you to be ready. You can all share the news on social media using @Gairdner Awards and tagging us @Gairdner Awards. So let's begin with the Canada Gairdner International Awards. These awards recognize outstanding biomedical scientists whose seminal discoveries have significantly increased our understanding of human biology and disease. The first Canada Gairdner International Award of 2026 is awarded to Dr. Wolfgang Baumeister. Dr. Baumeister is Director Emeritus and Scientific Member of the Max Planck Institute of Biochemistry, Martinsried, Germany, and Distinguished Professor at Shanghai Tech University in China. He's awarded for developing cryo-electron tomography, a method that visualizes molecular structures inside intact cells at nearly native resolution, creating a new way to study cellular architecture and revealing the inner workings of life at the molecular level. Let's hear a video about Dr. Baumeister.
I would say I think I saw more the potential of the method than the risks. I mean, the more serious people have said, well, this will not be possible. I think you're only going to destroy your sample.
Professor Wolfgang Baumeister developed cryo-electron tomography, an imaging method allowing scientists to see the molecular machinery of life inside intact cells. With the ability to visualize cells in remarkable detail, this revolutionary approach revealed how molecules are arranged and interact within their natural cellular environment. This technology is transforming how researchers understand the molecular interactions that underlie health and disease, opening new windows into the fundamental processes that sustain life.
The biggest influence had a teacher, a biology teacher in high school. My performance at high school was not that great. He gave me a project in plant ecology at the time. It was really great fun. I simply felt at that time it's much more fun to explore the unknown than to consume and take in what is known in the textbooks. One of the most important attributes of a scientist is to persist. Go where your curiosity is driving you. Don't be deterred by failures. I think in any career, I think there might be more failures than successes.
Congratulations to Dr. Baumeister. So the next 2026 Canada Gairdner International Award laureate is Dr. Jeffrey Kelly. Dr. Kelly is the L. Lucha Brown Professor of Chemistry, Department of Chemistry at the Scripps Research, La Jolla, California. And he's awarded for discovering the anti-aggregation drug tafamidis, the first effective treatment for the amyloid disease transthyretin amyloidosis, providing the first pharmacological evidence that protein aggregation drives neurodegeneration. And let's see the video.
I always thought about what we didn't know more than what we knew. And, after all, in science, that's what we're supposed to do, is challenge dogma.
Dr. Jeffrey W. Kelly discovered the first effective drug to treat transthyretin amyloidosis. a life-threatening disease that occurs when a healthy protein becomes unstable, falls apart, and forms harmful clumps in the body. Dr. Kelly uncovered how this degenerative process advances and developed a drug that stabilizes proteins, preventing them from breaking down and causing harm to the heart and nervous system. His breakthroughs have been shown to slow disease progression, transforming care for patients and opening new possibilities for treating neurodegenerative disorders.
I mean, the first time I met a patient, everything changed. Dementia in particular, which is one manifestation of having an amyloid disease, it was just so clear that my problems are minuscule compared to theirs. And, you know, it gives me great pride that we can extend their lifespan and give them a little bit longer quality time with their family. And I hope in a decade we can start to think about prevention as the main strategy. Up until I entered college, I didn't see myself as being a professional scientist. It is still today pretty hard to make discoveries in chemistry because it's a pretty mature field, but biology is completely wide open, and that was really exciting to me.
So congratulations again. And I'm happy to tell you that we have Dr. Kelly with us today, all the way from California, and we welcome him to say a few words.
Well, good morning. I grew up right across from Toronto on Lake Ontario, so it's a privilege to be home, as it were. I certainly wouldn't be here today without the sacrifices that my family made for my education. And of course, the creative contributions that the postdoctoral fellows and graduate students who trained in my laboratory made towards the discoveries that I've been recognized for. So it's really we, and of course it takes a village to make these kinds of discoveries that we just heard, for example, Wolfgang Baumeister's discovery. And of course there would be no drug tafamidis without the sacrifices of the patients, the physicians and nurses who do the clinical trials. It really takes hundreds of people ultimately to put a drug in front of the regulators for approval. I guess the most important message for the young people this morning is I hope a wonderful celebration like we're all privileged to attend today shows the younger generation how interesting and exciting it is to pursue science. It's wholesomely addictive and with luck on your side, can lead to benefits for society that are pretty easy to appreciate. Of course, there are many more failures than successes, so one has to persevere through them. It's such a privilege to join the Gairdner family, and I want to recognize the accomplishments of the foundation and getting young people excited about science. I'm really looking forward to our week in the fall. I didn't really grow up thinking that I wanted to be a scientist. I was really curious about how things work and really appreciative that my family was tolerant of me taking things apart and not knowing how to put them back together. Eventually, I learned how things work, although it was an expensive process for them. My tip for young scientists, I guess, is to be a closet contrarian, or to be really skeptical about how things work or how we perceive them to work. If I've learned anything in my career, it's that even when we think we understand a disease really well, we still can be substantially surprised when we get into patients. So be suspicious, be thoughtful, and most importantly, have a lot of fun at pursuing science. Have a wonderful day, everyone. Thank you.
Thanks, Jeff. That was great messages for everyone. The final recipients of the Canada Gairdner International Award is a trio this time, and the trio are Professor John Yates, Dr. Rudy Abersold, and Dr. Matthias Mann. Professor Yates is the John Lytton Young Endowed Chair and Professor at the Department of Integrative Structural and Computational Biology, Scripps Research. So Scripps is picking up 2 awards this year. Dr. Aebersold is Professor Emeritus, Molecular Systems Biology in the Institute of Molecular Systems Biology and the Department of Biology at ETH Zurich, Switzerland. And Professor Mann is Director of the Department of Proteomics and Signal Transduction at the Max Planck Institute for Biochemistry in Martinsried, Germany, who also pick up 2 awards this year. So we've got pairs coming through. Together, those threesome have been awarded for establishing the foundation of modern system proteomics through transformative innovations in quantitative protein measurement, mass spectrometry technologies, and computational analysis, which is really the fundamental discoveries that are having impact across all of biomedical science and health research. So let's hear a video about their research.
I was always interested in how things function, even as a kid. Science is a field which thrives on interactions.
I think a lot of people my age got chemistry sets as a Christmas present or something like that. I really enjoyed taking chemistry classes. And then luckily for me, I didn't get into medical school. You know, sometimes when a door closes, another one opens.
Originally, I was very interested in more abstract problems, so I studied physics and math, and I thought, okay, we can not only crack these abstract problems, but we can make a real difference.
Doctors John Yates, Rudy Aebersold, and Matthias Mann built the foundation of modern proteomics, the large-scale study of proteins that power life inside our cells. They developed technologies that allow scientists to identify and measure thousands of proteins with extraordinary speed and precision. This transformed protein research from a slow, fragmented process into a powerful systems-level science. Proteomics is now central to understanding disease, accelerating drug discovery, and advancing precision medicine around the world.
And so we totally disrupted how protein biochemistry was practiced, and that these methods and so forth have been so widely adopted around the world. And that's kind of awe-inspiring.
So this is an exciting convergence of these developments in AI and these developments in this proteomic field where we now can measure not just the proteins but their state. And if these 2 fields converge, I think this would be an extremely exciting prospect.
It will be going into the clinic, into hospitals, and that people can routinely be measured by proteomics technologies. So I think the next 5 years will be as impactful as the last 30 years put together.
Great. Congratulations to the trio. And I'm happy to say that we have Professor Yates with us from Scripps today. So Jeff came in directly from San Diego. John came in from Pittsburgh, where he was attending a meeting, so we've brought them all together. And John, welcome to the stage.
Good morning. Glad I could make it. I was a little skeptical of the flights. I want to express my deep gratitude to the Gairdner Foundation for this extraordinary honor. The Gairdner Awards have long recognized discoveries that have transformed biomedical science, and I'm humbled to be included among the remarkable scientists who have received this recognition, and especially with my colleague, Jeff Kelly. My work is centered around the characterization of proteins, the molecules that do most of the work inside cells. Yet for many years, studying proteins at scale seemed nearly impossible due to the complexity of biological systems and the technical challenges of protein sequencing. As far back as my graduate studies, it became clear to me that if we could solve the problem of accurately and automatically interpreting tandem mass spectra of peptides, we could open the door to entirely new ways of analyzing proteins. I still remember the exhilaration of the first successful data analysis. It felt like a true turning point, revealing a new path for protein sequencing and showing that this vision could, in fact, be realized. But direct sequencing of complex protein mixtures would require more than algorithms. It demanded new instrumentation. I was fortunate to have collaborators at Finnegan, now Thermo Fisher Scientific, who are willing to listen to what must have sounded like a very ambitious and perhaps crazy idea at the time. But these advances helped drive a fundamental shift in the field, from studying one protein at a time to identifying thousands in a single experiment. That shift laid the foundation for what we now call proteomics. Today, proteomic technologies are woven throughout biomedical research and have helped enable entirely new areas of science, such as chemical proteomics, bioorthogonal chemistry, systems biology, and large-scale interactomics. I want to thank my wife, Claire, and my sons, John, Connor, and Kenan, for sharing and supporting the journey. I also want to thank the many friends who have made the journey joyful, stimulating, and fun, and the many collaborators who have posed important biological problems that have challenged and expanded the technology. And importantly, this work was not done alone. I've been fortunate to collaborate with extraordinary colleagues, students, and trainees whose creativity and dedication made these advances possible. I'd also like to thank the University of Washington, where I started as an assistant professor, Scripps Research, where I am now, and the NIH, which has supported this research over many years. I'd like to acknowledge important mentors in my career, such as Robert Anderegg, who introduced me to mass spectrometry, Donald Hunt, my PhD advisor, who envisioned using tandem mass spectrometry to sequence proteins, and Leroy Hood, my postdoc advisor at Caltech, who envisioned large-scale biology. Finally, I want to thank the Gairdner Foundation for recognizing the importance of fundamental discovery science. I realize that deciding which scientific advances to honor is a Solomon-like exercise, and I'm grateful this year you chose to honor proteomics. As Freeman Dyson wisely said, new directions in science are launched by new tools much more often than by new concepts. I'm particularly honored to be here at the University of Toronto receiving this, because over 100 years ago, the discovery of insulin, the first miracle drug here at UT, helped launch Scripps Research Institute. Ellen Browning Scripps started the Scripps Metabolic Clinic over 100 years ago in San Diego to bring insulin to patients. And that led to the Scripps healthcare system and led to the founding of the Scripps Research Institute many years later. And so we are here in part because of the University of Toronto and the discoveries made there. So thank you very much.
So congratulations to the trio again. Obviously, we've heard across the international awards this year, we're looking at the fundamentals of how molecules work within cells, how proteins interact, the fundamentals of biology, looking at in new ways and new detail that is changing the way we think about how our own cells work and the impact on disease. So it's a fantastic group of scientists that I think will be a lot of interactions during the Gairdner Week when everybody is here. So now we're going to move on to the 2026 John Dirks Canada Gairdner Global Health Award. And I'm very happy to see that John Dirks is in the audience with us. This award is named after John Dirks, who was the previous president of the foundation to me. It was his vision that one of the areas we should really be looking at is not just research that has global impact in the broadest sense, but really research that takes place and has an impact in the global south. So really looking at research that impacts worldwide. And so this award is given to scientists whose advances have made significant impact on the health and well-being of those facing health inequities worldwide. This year, two laureates share this award. The 2026 John Dirks Canada Gairdner Global Health Award laureates are Dr. John Clements, who is Senior Scientific Advisor to the Director General at the International Vaccine Institute in Seoul, South Korea, and Dr. Jan Holmgren, who is Senior Professor at the Sahlgrenska Academy, University of Gothenburg in Sweden. And they're awarded for advancing the understanding of cholera and immunity, and for the development and evaluation of safe, effective, and affordable inactivated oral cholera vaccines that have enabled cholera control worldwide. So let's hear about their research.
One thing about our business, my circle of close friends, aligns fairly closely with my circle of close scientific collaborators. Friendship and science were never very far apart from each other, and were very, very sustaining.
It was only after I had started medical studies I got inspiring supervision and also a good basic knowledge. Aged just 22, I was hooked up on microbiology and immunology research. and have stayed in the branch since then.
Doctors John Clemens and Jan Holmgren transformed the fight against cholera, a life-threatening disease that spreads in communities without access to clean water and sanitation. They developed safe, effective, and affordable oral vaccines made from inactivated bacteria. These simple drops protect severe illness in individuals and can reduce the spread of cholera across entire communities. Oral cholera vaccines are now used worldwide in outbreaks in high-risk regions, helping to control the disease and save lives on a global scale.
I must admit I'm not a laboratory scientist. Fortuitously, I was able to link up with Professor Jan Holmgren, and my clinical and epidemiological skills and Jan's basic science discovery skills formed a perfect partnership that has really sustained the work that we have done over the past 4 decades.
My greatest happiness today, and also I think the great joy I feel, is the long collaboration Dr. Clemens and I have had, that these oral cholera vaccines, which we developed a long time ago, have finally come to use to save lives all around the world.
It was all a great adventure, and we saw results. Even though the political situation in the world was unwilling for many decades to translate those results into public health good, the public-private partnerships that really supported the work of Jan and myself were very sustaining.
So I suspect many of you who travel to places around the world have probably taken Dukoral, which is an oral cholera vaccine. But really what these two researchers did was not just develop it for you, but really continue to push very, very hard for many, many years to ensure that they were available in outbreaks in parts of the world that could not necessarily access these vaccines. So it's really made an incredible difference, and everything goes back to them. It's interesting you thought about friendship, because when I talked to Jan, he said that the two of them still have a phone call every Sunday morning. They chat about, life, the universe, and cholera, I guess. So, but really, it's a wonderful partnership, and I'm looking forward to seeing both of them here in Toronto. Finally, I'm going to announce the 2026 Peter Gilgan Canada Gairdner Momentum Award laureates. These awards are Canadian. They're given to mid-career investigators in Canada who in their last 6 active years have produced exceptional scientific research contributions with continued potential for impact on human health. And we're very grateful to the Peter Gilgan Foundation for their support of the Momentum Awards. And we're very happy to welcome Mary Federer, who's chair of the Foundation's Board of Directors, and Elizabeth McCallion, Executive Director of the Peter Gilgan Foundation, to this morning's event. So thank you again. The first 2026 Peter Gilgan Canada Gairdner Momentum Award laureate is Dr. Erin Phillips. Dr. Phillips is an associate professor in the departments of clinical neuroscience, cardiac science, physiology, pharmacology at the University of Calgary. And he's associate dean of innovation and commercialization in the Cummings School of Medicine at Calgary. And he's awarded for pioneering work that restores blood pressure control after spinal cord injury, reducing life-threatening complications, improving daily functioning, and transforming clinical care for people living with paralysis. Let's hear the video.
I was a very inquisitive kid, and my dad definitely indulged me and inspired me to be able to continue to ask why. With these games of why, you'd end up with very esoteric concepts like, well, why is there an infinity? And and then at that point, he'd say, okay, we're going to the library.
Dr. Erin Phillips is transforming care and redefining what's possible for people living with spinal cord injury. His research uncovered how damage to the spinal cord disrupts the body's ability to control blood pressure. He then developed an implantable neuroprosthetic device that continuously monitors blood pressure and delivers targeted stimulation to restore stability. His breakthrough therapy reduces reliance on medication and improves daily function, offering new independence and safety for people with paralysis.
People with spinal cord injury have about 6 times the risk of cardiovascular disease. This is about double the risk that smoking has for heart disease. Once you understand exactly how something works, you can quite, in a straightforward manner, bring that forward to a therapeutic with so much less doubt along the way. Fundamentally, science is what progresses our society, our healthcare system, our technology, and I think sometimes the reward network is on a little bit slower cadence, but the outcomes can be profound. You can improve health on a population level if successful.
Great, thank you, Dr. Phillips. Congratulations again. The second 2026 Peter Gilgan Canada Gairdner Momentum Award is given to Dr. Karen Maxwell. Dr. Maxwell is Professor of the Department of Biochemistry right here in Toronto at the Temity Faculty of Medicine. She's awarded for uncovering the molecular strategies bacteria use to defend against viruses, revealing how viruses known as bacteriophages evade those defenses, and laying the foundation for next-generation precision-phage therapies to combat antibiotic-resistant infections. Let's hear the video.
It's an amazing job. It's every day we're discovering new things, and these are incredibly bright and talented young people, and it's just so much fun to come in and work with them every day.
Dr. Karen Maxwell is uncovering the hidden battle between bacteria and the viruses that infect them. Her research reveals the remarkable ways bacteria defend themselves, from blocking viral attacks to activating viral sensors hidden within their own DNA. Dr. Maxwell has initiated a new generation of precision phage therapies, treatments designed to target dangerous, antibiotic-resistant bacteria without harming beneficial microbes, opening new possibilities for fighting infection and protecting global health.
As a child, I was always really interested in science and math, but as a teenager, I had a brother-in-law who died of cancer, and it was that actually made me decide that I wanted to do biomedical research and just moved into that field. A lot of what we do as scientists is challenge each other. We poke holes in each other's arguments, and that's what science is all about. And so I think, students need to have confidence in themselves, have confidence in their ideas, but also be resilient enough that when people point out problems with your science, that you're strong enough to withstand it.
We're happy to have Dr. Maxwell with us this morning, and now I invite her to the stage to offer a few remarks. Karen.
Thanks, Janet. So I was a graduate student here in Toronto, and I've been attending the Gairdner lectures every fall for over 2 decades. And it really is so exciting. Every year it's exciting to see the research that's being recognized, and it's particularly exciting, of course, this year to be recognized for my own curiosity-driven research. And I'd like to thank the Gairdner Foundation and the Peter Gilgan Foundation for their support of this award. And of course, the award is recognizing me, but science is a team sport. It's not done by one person alone. And there's a lot of people who helped me out and supported me along the way I'd like to thank. First and foremost, of course, are the staff and trainees in my lab, the people who are the hands that do the work, and a bunch of them are sitting right over there at that table. I think a lot of people don't realize, but the bulk of biomedical research that's done in this country is done by these incredibly bright and talented young people. They're in their 20s, early 30s, and they are the ones that are making these incredible discoveries. So again, thanks to you guys. I'd also like to thank collaborators and colleagues here in Toronto who have supported me, not just scientifically, but also personally in my journey in science. And of course, my family, some of whom are also here celebrating with me today. Their support over the years, they are without a doubt my biggest cheerleader and are always there asking me how things are going. And in particular, I would like to thank my husband, Alan Davidson. He's my partner, not just in the lab, but also in life. And, we have really built this research program together, and I couldn't have done it without you. Thank you.
Thanks, Karen. Congratulations to you, and welcome to all our laureates. You are now part of the Gairdner family. Really, as president of the foundation, it's a true pleasure to welcome 9 more remarkable individuals into the Gairdner family. It's a great community of researchers leading some of the world's most impactful breakthroughs in health-related science. Celebrating excellence, inspiring the next generation of researchers, fostering public trust in science are central to Gairdner's mission and brought to life through our national outreach initiative, Gairdner Connects. So throughout 2026, our laureates, these laureates, past laureates, other leading scientists will travel across Canada to engage with faculty, trainees, students, and the public, sharing their groundbreaking research, but also the stories behind it. And you heard a little bit of those stories coming through the videos. And if you want to explore what's going on in the Gairdner world this year, we invite you to follow us on social media, and you can visit our website, gairdner.org, and subscribe to our newsletter, which will keep you up to date on symposia, workshop, and other events that are taking place across the country. So now you know who the laureates are. Now they're from all over the world, and now we're going to hopefully bring them to life on the screen and join us in a live and online discussion. So I'm hoping that's going to happen. Meantime, yes? Oh, yes. Hello, everybody. Fantastic. Great to see everybody. Now I'm going to ask the current laureates who are here to come and join me on the stage as in real life, and we're going to have the opportunity for me to chat with them and ask them some questions. We also have high school students in the audience and a couple of high school student questions that will come up at the end of this discussion. And as every laureate knows, the high school student questions are the hardest. So please join me also. I think it's on wheels. Just for, okay, good. All right. Now, let me see. I can see on screen Dr. Baumeister. Can you hear us? Can we hear you? Can people unmute themselves?
I can hear you.
Yeah, good morning, or yes, good afternoon, Dr. Baumeister. And Matthias Mann, next to you. Welcome.
Thank you.
Nice to see you. Dr. Holmgren, welcome. Going round, Jan Clemens, joining us from Korea, Dr. Clemens.
Hello.
Good morning. Rudy Ebersold, welcome.
Good morning.
And Aaron Phillips, welcome. So we've watched your videos. I hope you've been watching online. Everybody knows a little bit about what you do. And I'm joined by 3 more laureates on the stage here. But I do have some questions, so we can explore a little bit more about your research and your background. You have been primed, so this shouldn't be too much of a surprise. I'll try to keep to the script, but sometimes we might wander off, who knows. So I'm going to start with Dr. Baumeister. So your development of cryo-electron tomography has really been a game changer in enabling visualization of molecular interactions directly in cells. What led to the breakthrough? What were the key moments where you knew that this was going to work? What were the key moments in your journey?
I think there was not a single key moment. I think it has been a very long journey, as a matter of fact. I think in mid-1980s, we were studying the molecular machinery of protein quantity control in cells. Our focus was on the proteasome, the principal molecular machine for the selective destruction of proteins that were either no longer needed or had become dysfunctional. So I think we used the established tools of structural biology, X-ray crystallography, single particle electron microscopy. With some success, for instance, we were able to identify the long enigmatic active site of the proteasome, but many mysteries. We felt very strongly that we might unveil some of these mysteries if we could obtain molecular resolution images of the proteasome, not only in isolation, but also in its functional environment as well. And one option appeared to be electron tomography, but there was a great deal of skepticism in the scientific community. The main reason is that biological material, especially embedded in ice, as needed in order to preserve them in a lifelike state, is extremely radiation sensitive, and tomography requires multiple exposures to the electron beam. So it was clear to us that I think it could only work if we could keep the electron exposure within tolerable limits by automating data acquisition. Now that was, very difficult at the time because microscopes were then still, designed to be operated manually. So it was a matter of hardware and software development at the time. I think a breakthrough was achieved in 2000 when we obtained for the first time cryo-electron tomograms of a cell grown on an EM grid, showing in remarkable clarity, several macromolecular machines inside the cell. That was, also by science then labeled as the breakthrough of the year. So that was the point in time when people began to believe that it could become something useful.
So so many steps along the way until it really comes to fruition with many different parts coming together to make it happen. And it's certainly a technique that's being widely used these days. So my next question is, what's the future?
Well, the future is, to push, of course, the boundaries of cryo-electron tomography, not so much in terms of resolution, but more in terms of comprehensiveness. I think what we want to achieve is clearly to obtain, to visualize, basically the entire molecular inventory of cells. The visualization is not the challenge. Tomograms are pretty holistic images of cellular environments. The challenge is the identification of all the densities, because what we image, it's a label-free method. What we image is the spatial modulation of density inside our sample. And so the challenge is to identify all these densities. In a cell, there are millions or billions of densities which need identification and annotation. I think that is a computational problem. I think it can be done by template matching. So if we have a template structure, we can compare it and, calculate a similarity score. So that works quite well. We have shown that it works in principle, but to scale it really to millions or billions of molecules, that is still a challenge.
So I think there's going to be some overlap in where you're going with where we're seeing proteomics and everything else going. The whole interactome of the cell is becoming more and more something we have to explore. So multiple technologies converging in really giving us amazing new insights into how cells work. So my next question, my next person, I'm going to the stage, and I've got Jeff in my sights. And the question I have, because we talked about it, it's in the video, you've talked about it, but I'm not sure I fully understood until I read about your research, the disease that started all of this for you, which is transthyretin amyloidosis. What is it? How did you get interested in it? And what does your drug actually do?
So the amyloid diseases are diseases whereby a protein undergoes a shape or conformational change
allowing the protein to misassemble into myriad structures, including amyloid fibrils, for which the diseases are named. So our vision in the early days was that we could intervene in these diseases by inhibiting the aggregation of newly biosynthesized protein. That wasn't a very popular idea because the thinking was that the amyloid fibrils having a specific cross-beta sheet structure between cells were the main drivers of the pathology. But it always made sense to us on a purely sort of biochemical perspective that aggregates that had the ability to access multiple biologies would likely be more of a driver of pathology. And so the drug we discovered binds to the newly biosynthesized protein and prevents it from aggregating. And we're fortunate, with the help of many physicians, nurses, and particularly the patients, to show that the aggregation of newly biosynthesized protein, even in the absence of removing amyloid fibrils, is sufficient to slow down the progression of human amyloid diseases.
So obviously, people are aware amyloid comes into people's brains thinking about Alzheimer's and other diseases as potentially amyloid diseases. So obviously, what you've done with this disease leads you into potential applications in other areas, including Alzheimer's?
Sure. I mean, Lars Landfeld in Sweden discovered an antibody that removes amyloid fibrils and also prevents the aggregation of newly biosynthesized A-beta. And when used in early stage Alzheimer's patients, that certainly slows down disease progression. And so I'm optimistic that aggregation modulators in general will be very useful. In fact, now we have 10 medicines that modulate protein aggregation for diseases like hereditary ALS, Alzheimer's, and transthyretin amyloidosis. So that's not to say that we don't need new mechanisms. We do, but we've at least got started on ameliorating these disorders.
Yeah, fantastic, that's great. Okay, so let's move on. And now I have the proteomics trio, and I've got several questions here. You all came at this in sort of from different directions, but it's all converged over the years. So the work that you've done, the three of you, has really been foundational to allow you to progress from low throughput analysis of a few proteins at a time. And I was saying yesterday, you know, when we first had mass spec groups in Toronto, I collaborated with a number of groups where we were trying to do mass spec on multiple proteins in different tissues, and they'd always want lots of material. And I work on early embryos, and I'm like, I can't give you lots of material. And so there was a number of issues that came into play that the three of you together over the years have really solved, to be able to take a few proteins, even hundreds of proteins, but requiring lots of tissue, to a situation where small amounts of tissue and many, many proteins. So really proteomics at a whole different level. So my question is key inflection points in that journey, and maybe I'll start with John since he's sitting next to me. Key inflection points in the journey for you.
So what has clearly been important is the interactions that the three of us have had with instrument manufacturers and helping guide them on where the technology should go and what's needed to do these things. We needed better sensitivity, we needed faster scan speeds, we needed better dynamic range. And so that inspires them to develop these new technologies. And so that's allowed us to continue to advance and drive the field forward using better computational methods, better sample handling methods, better approaches for new concepts for how we would want to study proteins and so forth. And that's allowed us to drive into areas that we probably never thought we would be able to get into, like Matthias has done spectacular work in the single cell field, single cell analysis. I'm really interested now in the 3D conformations of proteins in vivo. Rudy has done spectacular clinical analyses using proteomics techniques. The blood is like the black hole of proteomics because it's got this enormous abundance range of proteins. And so just through all these developments we've been able to achieve over the years has allowed us to really push the field forward.
Great. So Rudy, you spent quite a lot of your career actually in Canada, UBC. Was that a critical time period for you in your journey?
It's absolutely a critical journey, time point in my journey and position. And in that context, maybe worth mentioning that I come from biology and from protein chemistry. So before I came to Canada, I never worked with a mass spectrometry instrument. And it was actually a program, a Canadian government program, Science and Technology Canada, which allowed me to acquire a mass spectrometer with a lot of help from the Canadian company SciEx. And this was an absolute inflection point because the chemical methods we used before to study proteins and to study protein sequence simply did not scale. And so I am extremely grateful to the Canadian government, to SciEx, and to UBC that gave me the opportunity to really go through this inflection point and learn about mass spectrometry and then start to using it to answer biological questions.
Great.
So maybe I can add that an additional, I'm coming from biology, the biology side. So for me, the inflection points were, of course, technical because they are enabling, but I was always driven by challenges posed by biological systems. And maybe we'll come later in the discussion to discuss where we see, the three of us, where we see the field going.
Right. So Matthias, from your point of view, inflection points?
Yes, but there have been quite a number of them, and there continue to be. So I was fortunate to be involved in electrospray, which even made it possible to put the biomolecules in the gas phase. So we were in a very small, underfunded group at Yale and sort of stumbled upon the electrospray, and that's kind of the basis of biological mass spectrometry now. And then I've been going back and forth, as typical for scientists, or should be. And then I was at EMBL, where I also got immersed into the biology in Heidelberg, so see where we can really use this for fundamental biology. And then I was in Denmark at a medical school at the Max Planck with more resources, where we could really, like Rudy, build this out to a systems biology level. So, and trying to understand the cell, not only at the genome and transcriptome level, but also what we would say is the real actors in the cell at the protein level. And that also, I was very fortunate again that all these things build on each other, and the journey is not over. So it's really said already. Now our field is merging big time with AI, and that's totally changing the game again. So we can actually deal with these huge data amounts that we can now acquire, and already a disease has been solved by proteomics that was totally incurable before, and I think that's probably just the beginning of it.
So over the years, a build-up, but this is an area that certainly is not finished. There are many more things and many discoveries to find, so fantastic. Question about the technology. So I ran a research institute, and every year it seemed to me that the people doing the mass spec would come and ask for a new, more expensive machine. Are we seeing an opportunity where mass spec will become more cheaper, faster, and will it turn into a clinical use? Any of you can answer that.
We asked for that.
Okay.
Yeah. We asked for mass specs that cost no more than a car.
Yeah.
But they probably don't make as much money on that. So there's the Tiffany model of business, and then there's the Walmart model of business. And I think they adopt the Tiffany model of very expensive instruments. And it's not every year, it's every 18 to 24 months, something new comes out. We got to go out and raise the money to go out and buy it. But I don't know, that's the way it is.
Let me push it again to the other two. In the clinical lab, do we see proteomics being a tool that can be used in the clinical lab?
Yeah, that's definitely our goal. And as Rudy said, he started with Sykes, and we now, which is in Toronto actually, and we work very much with them, and they've just come out with an instrument that's not like, it doesn't cost as little as a car, but it does cost much less and is much more robust than current high-end instruments, and I think that can go in a clinic. And for small molecules, for drugs in the blood, there are mass specs in the hospital already. Yeah. So I think there's no reason why this shouldn't be the case for biological mass spectrometry, in a couple of years as well. So I think it's only a question of time.
Great. Good. Thank you. Good messages for people in the audience who are running labs or running clinics or running institutes. So I'm going to turn to John Clemens and Jan Holmgren. So you've worked together, the two of you, and I know you're still very closely interacting with each other for over 40 years to transform global cholera control, creating oral vaccines, ensuring they're affordable and effective. And I think this is really such a lifetime commitment to this study. It's really amazing. So I know you're very close. How did you meet? How did the meeting occur? How did this start, your somewhat unusual partnership?
I guess, Jan, can I take this one? It began in 1983. At that time, WHO had come to agree with the then fairly new notion that for cholera vaccines to be effective, they had to be given by mouth, not by injection. However, an oral cholera vaccine had never been tested for actual safety and efficacy in practice, and WHO decided to support the world's first trial of an oral cholera vaccine in Bangladesh at an institution called ICDRB. But the problem was there were several oral cholera vaccines in development, and one had to be selected for the trial. So the WHO convened a meeting at the Shoreham Hotel in Washington, D.C., which I attended because actually quite independently, I had been recruited from Yale to lead this trial of whatever vaccine it was going to be in Bangladesh. And Jan's vaccine, which later became Dukoral, won the Derby at the meeting, and that initiated our friendship and collaboration, which, as you mentioned, has continued for 4 decades.
So great, that's fantastic. And obviously the impact on the cholera area of your friendship is very important. And we've heard that mentioned by others. Friendship is part of being a scientist. So there's this concept that, scientists work alone and don't talk to people. It's not true. Science is a collaborative social interaction, and it's absolutely essential for making science work. It's the fun part about science. So, we're talking vaccines here, so let's talk a little bit about other vaccines and your viewpoints, both of you, on where we stand as a world, I guess, in making sure that as we face new pandemics or we face existing diseases like dengue, HIV, and others, malaria, how close are we to being able to have effective vaccines, but also importantly, making sure that those vaccines are available in the global sense? Where do we stand today? Who wants to go for that? Yeah,
I think Jan will take that.
Well, you mentioned dengue and HIV, and if we take dengue, it's a bit like for cholera. There are vaccines which have been developed and are effective, and there the main problem is not scientific, but more one of supply and access to make these vaccines which are available accessible to those who are in the greatest need of it. So here, although of course for dengue as well as for cholera and several of the others in the same situation, better vaccines can be made, but the main problem will be in health politics and finding enough money to support the access of these available vaccines and new vaccines, of course, to those who need it. For HIV, on the other hand, the situation, as you know, is that there is no effective HIV vaccine. So there the science remains to develop one effective, truly effective HIV vaccine. And once that has been done, of course, then the situation will be a bit the same as for dengue and cholera, how to make such an effective vaccine available to the poor people. Again, more politics and more the financing situation than science.
Yeah, so obviously politics and access to the funding, the cuts in some of the funding that's given by a number of governments, not just the US government, have impacted many aspects of global health. And in the vaccine area, it's not just the removal of funding, but we have the whole issue of vaccine hesitancy, vaccine misinformation that is making it very difficult for new vaccines to actually be developed and distributed. Any suggestions on how best to counter vaccine misinformation?
Well, yeah, vaccine misinformation clearly is the jet fuel that helps drive vaccine hesitancy. And the problem of vaccine hesitancy is enormous and growing globally, and it really threatens the entire public health vaccine enterprise. Of course, in dealing with vaccine information on an individual level with a patient or a small group, it's obviously advisable to deal with it in a factual and respectful fashion. And I might add that our vaccine establishment is not always as respectful as it should be in this regard. But that's really, I think, that's the Band-Aid. The real problem, I think, underlying all of this is growing public distrust in institutions and in science. And I think to tackle vaccine hesitancy, as well as a variety of other major problems, it's that fundamental issue that needs to be addressed.
I think that's an area that we've talked about here and we talked about in many contexts, this mistrust of science, and mistrust of evidence, mistrust of really how to study the world in a rational way. And as scientists and as institutional leaders, we have to stand up, we have to be counted in whatever situation we can. This is an ongoing discussion that we're not going to complete today for sure, but I appreciate your insights. I'm going to turn now to Karen, which is perhaps in the future you'll be facing a pushback against what you're doing as well, but at this point your research is very much at the discovery level. You're trying to find how bacteria fight their viruses, and it's really exciting. So everybody knows about CRISPR, but you're defining new ways that bacteria actually have their own immune system. So can you tell us a little bit more? I don't think we got a lot from the video about exactly what is going on there. People probably don't know, bacteria have viruses. Viruses are everywhere, and bacteria have to have a means of fighting off their viruses. So can you just give us a little bit more understanding?
Yeah, so as you said, bacteria have their viruses, and they have immune systems like humans do that help to fight off these viruses. And we're really interested in figuring out all of the different ways that bacteria defend themselves against these viruses. And there's some really important reasons for that. First, of course, is that we can discover potentially new biotechnological tools. So CRISPR-Cas and restriction enzymes are both bacterial immune systems. And they were discovered through basic curiosity-driven research. And people figured out how to take these systems out of bacteria and use them in a test tube. And really, these are what have enabled modern molecular biology and genome editing. And so further study of these systems could reveal really important new biotechnological tools. The second reason is to deal with antibiotic resistance. So antibiotic resistance rates are rising at an incredibly rapid pace. And drug discovery is not keeping up with increasing rates. And phages, these natural viruses of bacteria, can actually be used to treat infections in human patients. And there are compassionate use cases that are ongoing at this point in time. But we can't just be fishing these phages out of the sewage, which is where a lot of them come from. Really, we need to be thinking about using phage therapy as precision medicine, and that really is the path forward. If we don't have effective antibiotics or antimicrobial treatments, all of modern medicine falls down. We can't do transplants, we can't do chemotherapy. And so really the basic discovery biological research of the type that we do in my lab is aiming to figure out all of the ways that bacteria defend themselves against these viruses, and also all of the ways that the viruses evolve to overcome those defenses. So in the long term, what we'll be able to do is sequence a bacterial strain, figure out here's exactly how we create this precision medicine, and then design it using synthetic biology and be able to treat antibiotic-resistant infections.
So it's very exciting. It's frontier work, I would say, at this point. But on a more personal level, Karen, I know that you're really invested in supporting the next generation of young scientists, something we've talked about today, particularly women and underrepresented groups. So can you tell us about what practical steps you personally are taking, and what other steps might we all take?
Yeah, I think, there's still, at this point in time, women are still underrepresented in science. So we've made great strides in getting girls into STEM, certainly in biomedical research. At the undergraduate and graduate level, we have equal numbers of male and female grad students. And in medical schools now, most classes are more than 50% female. So we've made incredible progress there. But past that point, we lose women. And really, to keep women in science, I think there's a few things that we can do. One important thing is visibility. You can't be it if you can't see it. And, I think, look around, we still, at the highest levels, women are still really underrepresented. I was at a meeting 10 years ago, and it's in a field that has a lot of high-profile women researchers, and it was the third day before a woman speaker came up. And when her talk finished, a graduate student in the audience stood up and said, "I am so glad to finally see a woman up there." And, it really struck me, and I looked at the speaker list for that meeting this year, 20% female. So again, I think visibility is one thing that's really important. And I think in some ways also there still are systematic barriers in place. Certainly to become an academic researcher, the really hard times when you have to work really hard and the really poorly paid times are when you're a grad student and a postdoc and a beginning faculty researcher, and those are really the prime childbearing years. And I think that's what keeps a lot of people, a lot of women, from advancing in science. And
the structures that we have in place haven't changed since the 1950s. To become an academic researcher, you do graduate school somewhere, you're expected to move away somewhere else, away from family support, away from your friends' support. And I think we need to be thinking more about how do we have some positions that allow women to either stay in one place and advance. How do we change the structures or the perceptions on hiring committees at academic institutions? But also, are there some kinds of special fellowships that we can put in place that allow women to have research-intensive programs, but without a lot of the other administrative things that go on when you're a beginning faculty member?
Okay, so obviously you've thought about this, and you have a number of important proposals that need to be pursued. So you now have a stage on which you may pursue these. And she's holding me to account, and the Gairdner Foundation to account as we go forward. And as people know, that from my point of view, one of the things that is incredibly important for the Gairdner Foundation is that we are open and we encourage diverse nominations. We want to see more diversity in all of our awards. We can do that through the nomination process. When it comes to the final decisions, sometimes it falls out that we have predominance of males. We have had more females. We need more nominations. We need everybody to be thinking. And any of you who, nominations are open. Anyone can nominate, and it's extremely important that we dig deep to ensure that we have not just women, but also diverse people represented in the awards program. But that's another discussion for another day as well. So now, sorry, Erin, I know you're still waiting. So now let me turn to Erin Phillips. So your work is interesting because, you do sort of span the basic science to clinical in one person and one program, one discovery and one application in a very short period. So congratulations on that. So tell us about the biological basis of what you've been doing, the biological control of blood pressure, and how you worked out this connection with the spinal cord and then translated it into an implant.
Yeah, thanks for the question. One thing to note, I think I mentioned it in my video, is people with spinal cord injury and other neurological issues die of cardiovascular disease at incredibly high rates. And historically, this has been a huge unaddressed patient priority. At a fundamental level, after honestly over 5 years of deep foundational science, we discovered that areas of the spinal cord injury, which we call the hemodynamic hotspot, are super powerful for controlling hemodynamics and blood pressure. And this is important because although after spinal cord injury, the brain loses its ability to regulate blood pressure, and those communication lines with the cardiovascular system are disrupted, the circuitry below the level of injury is alive. It's just disconnected and dormant. So one of our key findings was identifying what we've called that hemodynamic hotspot in the spinal cord. It's in kind of the lower thoracic region. We used a number of sophisticated techniques of highly specific viral tracing, functional experiments, et cetera. And we found that this is a key region where a small population of neurons has a very disproportionate influence over hemodynamic control, and that's largely due to the massive amounts of neuronal divergence of the sympathetic nervous system, where limited primary original neurons exert huge amounts of control over end organs, like the blood vessel network. So what that gave us was this target. After all this science, we had this potential target for a therapeutic. We could deliver very focused neurostimulation to re-engage that hotspot and stabilize the system. And then that's where really the translational journey began, which is a whole other kind of field of science and a lot of learning on the fly, backed by a lot of commitment from my team. But that's what enabled us to develop this neurostimulation implant for patients that translates into less fainting episodes, clearer cognition, better engagement in social and professional activities. And likely we're doing these experiments now, but it appears like likely much greater, a huge reduction in the risk of death and disease. I'm also part of the journey. We partnered with Onward Medical, a publicly traded company, and we're in trials right now with the goal of securing regulatory approval to bring this therapy to the community. So the ARC was identifying this kind of critical control node in the biology and then building a clinical grade technology to selectively modulate it. So that was the journey.
It is quite an incredible journey, and it makes it sound sort of easy, but that's not easy because you really had to build active collaborations between the lab, the clinic, and then the company. Biggest challenges along the way?
Yeah, I mean, there were obviously lots of fields coming together and learning to kind of speak different languages between experts in different fields. One, I thought about this quite a bit, and probably one core challenge for those interested in truly translational work is that different fields define success very differently. In foundational science, success is often about deep mechanistic discovery and increasingly sophisticated technical approaches. You're really at this level of deep science. In the clinical world, success is more pragmatic. It's about robust, reproducible effects, gold standard outcomes that really matter to patients. So the intercommunication between fields comes during that translation, and something that can be scientifically compelling, it may not be compelling to justify a surgical intervention. So when we moved into clinical trials and working and convincing clinical collaborators to come on board, a major focus was ensuring the effect was not just real, but durable and clinically meaningful. We had to be confident that the benefit outweighed the risk of a surgical implantation. And that meant designing studies and technologies that, prioritize pragmatism and being very, very disciplined about when the science was ready to take the next step.
And of course, the patient voice is very important. And again, I think we've heard that throughout these studies. In the end, what people are working on is going to impact patients, but you need to understand what the patients value. And I think that's why your research is particularly interesting, because for the patient, controlling blood pressure is incredibly important. The average person might think that the person with spinal cord injury is more concerned about being able to walk, but if they can't control their blood pressure and live a normal life, that's a really quality of life issue. So very important work. Well, I've had a good conversation. I hope everybody else has enjoyed the conversation that we've had today. Now we're going to ask the high school students. There's a couple of high school student questions coming up. We won't have time probably for all of you to answer both questions. You can put up your hand if you want to answer the question, or I may point at people as well. So can we have the high school student questions, please?
Hi, my name is Crystal, and I'm a student at TKS currently exploring how gene editing can be used to improve inflammatory skin disorders. My question is, what is one scientific problem you hope someone in my generation will solve that hasn't been solved yet?
Okay, one scientific problem for the next generation that hasn't been solved yet. Who would like to take that one? Jan.
Well, of course, there are many. From my perspective, I'm basically an immunologist. And the immune system has two equally important functions. One is obviously to protect us against infectious diseases. But equally important is that we can become tolerant against things which are causing autoimmunity or allergic reactions. And while we have been fairly effective in developing effective vaccines against many infectious diseases, as you know, we have been less successful in developing useful vaccine-like treatments against autoimmune diseases, which are many of the more severe chronic diseases like type 1 diabetes, rheumatoid arthritis, multiple sclerosis, and perhaps even atherosclerosis. I worked quite a long time on this. I thought we would have come equally far as we have done with some of the vaccines. We have successful results in mice, but it certainly remains a new generation to develop this into practical medical treatments, creating those vaccines that can specifically tolerate against many of these chronic diseases I mentioned about. So that would be my contribution to a good topic.
Okay, anyone else? Matthias. Matthias,
Yeah,
and then Rudy.
Yeah, just my 2 cents worth is aging. So I think aging is the ring that rules them all. So if people in your generation could not age, then they would live at least 1,000 years. Yeah, more and more we realize that everything comes together in aging, so it's very interdisciplinary. It will need new methods that are not even developed yet on systems biology and proteomics even, and all that. So if I was a young researcher, which I'm not, then I would go into aging now.
Okay, aging. Rudy?
I think this is an excellent question from a high school student because it has a long-term perspective, and I believe that with the advances that have been reached in understanding by how biological systems work, cells, organs, whole organisms, that through the convergence of some of the technologies that are being recognized here, convergence of them, for instance, cryo-EM and proteomics, together, of course, with computational methods will allow us, the scientists at that time, to understand how a biological system reacts to a challenge. The challenge could be aging, challenge could be radiation, challenge could be some other disease, and to take measures that are knowledge-based, based on mechanistic understanding of which processes are perturbed. I think this was for a long time not on the horizon. There were attempts out of the genomic project, the genome project, to make these predictions already. And I think this disregarded the complexities of the molecular entities of a cell, and that through techniques we've been discussing here, and of course many other techniques, this goal of understanding enough to make predictions that are meaningful and can be interfered with is on the horizon.
Okay, I think those are all good. Okay, one more from John.
I'm at the risk of being overly pragmatic. The foundation that enables all this good stuff to happen is public trust in science. And I frankly think that we are just winging it now in addressing this problem. And I think that fundamental advances and approaches to really resolving this issue will be critical.
So that's, I think, a great way to end that question, because obviously it is the next generation who really have to have trust in science, and our role is to make sure that that's embedded in everything that they see, do, and think. The next question from the high school student.
Hi, laureates. My name is Sidi. I'm 16 years old, and I'm an innovate at TKS. My question for you is, what was the most unexpected obstacle between your research and the impact it was meant to make, and how did you navigate it?
Okay, unexpected obstacles. We've talked about challenges. Let's try something unexpected that happened in your journey, in your research. A sudden surprise. Rudy?
Yes, so again, I think this is a very interesting question. And I think for me, on this journey, the most unexpected obstacle was the assumptions that were prevailing in the field. And we all learn, if the high school student is probably learning the basics of biochemistry and molecular biology, we learned that a dogma in molecular biology is that one gene makes a protein, makes a function. And we learned other things which have really dogma level status, and they are all not true, but they're all too simplistic. And I think it is really important to challenge these assumptions that are in the field and not just operate and plan experiments and plan studies within the prevailing dogma that may be taught in the basic biology lectures, but to really try to see what are the constraints and what is the reality for these assumptions in the present time, so that the experiments and the studies can be adapted to the modern constraints and not the ones which are in textbooks. Not that these are wrong, but I think they were established at a time when science simply was different than it is now.
Yeah, so don't be afraid to challenge dogma, and it's been something that all of our laureates have been challenging all the way through, I would say. Aaron.
Yeah, thanks. I think this is a terrific question, so thanks for submitting this one. As a neuroscientist, delving into kind of the clinical world and clinical trials, one super unexpected obstacle was realizing how hard it was for our patients to participate in research. And maybe that's also, a little bit pragmatic for this group, but we tend to focus on the science and the technology. But for many people, especially those with disability, just getting into the lab, completing assessments and staying engaged is a very significant and expensive in terms of time and money problem. It's a huge burden for these folks. So I don't know the exact solution is what the final solution is going to be for these types of issues, but for us, it kind of forced us as a lab that develops technology to rethink how research is done. And along the way, we started developing tools to bring research into people's daily lives instead of asking them to come to us. So that led us to developing a platform that we're launching across Canada that allows us to collect data remotely, biological data and patient-reported outcomes, and it makes participation way less burdensome and gives us more continuous real-world data. I'm sure there's other options that will emerge over time, so maybe I'll plant the seed as something to solve in the future also for the previous question. Thanks.
Great. Any other final comments on this one? John.
So I have a very specific one. That was particularly when I was junior faculty, and we had just developed this database searching process, and we were trying to get it published and also get some research funding for it. And submitted the grant, got a really terrible score because one of the reviewers thought that, well, we were doing it all wrong and we needed to do it his way. And the way that he described to do it, even 30 years later, still wouldn't work very well. But also trying to get the paper published, and I sent it to a journal where I thought the editor of the journal would appreciate the work, and he rejected it. But he gave me some really bad advice. He said, "Oh, just have the next editor say, call me up, and I'll tell them who the reviewers were, and that'll speed up the process." It wound up that being terrible advice because the person who was reviewing it really didn't like the paper and kept rejecting it. So it was rejected 3 times before it got submitted. But the important lesson out of this is that I then took it to my PhD advisor, my former PhD advisor, and I said, "I'm struggling to get this paper published." He said, "You got a copy?" I gave it to him. He walked it over to the editor of one of the mass spectrometry journals, gave it to him and said, "You need to publish this." And he said, "Oh, this is great." And he published it, and it's the most highly cited paper in his journal by a factor of 6. And whenever anybody brings up the paper, he just smiles like a Cheshire cat because he picked this up. So the important thing is that you got to be tenacious in science. People are going to be telling you things that they believe, and they may not be true or not. I think Max Planck said, dogmas don't die until all the proponents die off. And so you get that sort of behavior going on. But it's also what's important is to have good mentors who can have your back and help you with problems like this when you're trying to deal with things.
So some stories that I think are common across many scientists, but there's the story about, the highly cited paper that has been rejected many times is very common. And that goes back to this challenging dogma, because when you come in with something new, the system is not ready for it, and the journals are not ready for it, but the people who get our awards are the people who are making those challenges and making a difference. So thank you to the high school students. That was fantastic. And thank all of you on screen, here, streaming around the world. Thank you for joining us today. I think it's very clear that we've had great stories. Our laureates highlight the profound impact that scientific discovery has on improving lives around the world. They remind us of the importance of sustained investment in scientific excellence for the future of Canada and the global community. And we encourage you to carry this message forward. So, to everyone here and those joining us online, thank you for being here, and we wish you the best of the day.